Hey, you're a human. I'm a human. We're different, but I want to be more like you. How can this happen? So basically, what happens is that we're made of trillions of these tiny little living things called cells, which pile up to make a bigger thing that basically uses cells to do the same thing cells do, only bigger and slower. It's weird. Anyways, you're a big pile of cells, and let's say you want to change something about yourself because it sucks and it's killing you on the inside. You can't do anything about it though. What you can do is change it for your kid. You see, these cells have a super secret set of instructions in them that tells them how to make more of themselves. That's pretty cool, I guess. Now, to change it, you need to sneak in and take the instructions and replace them with the ones you want the cells to make, so the whole pile will change and become the kid, except, less likely to die quickly. A lot of very smart people argued about the best way to break into cells and steal the instructions. But none of their ideas worked because they decided to just change what the cell was making, not what told it to make things in the first place. Then some other really smart people said, hey, what if we use the thing inside the cells to cut the cells and replace the things inside of them? And their other smart friends said, that'll never work. And then some really closed-minded right-wing people who probably needed to be smarter were like, no, don't do that. Genetic modification messes with the bodies that God gave us and can probably lead to world domination. But who needs ethics? CRISPR still worked out anyways. So let's recap. CRISPR became CRISPR. It became technology. Not just any technology, but genetic technology. Instead of editing the soup around the instructions or the proteins that soup tells things how to make, it edits the DNA itself. It's super effective when it comes to removing bad stuff like genetic deformities and other nasty medically incurable things, like HIV, or autism, or death. So, how does the soup cut the DNA where you want it to instead of leaving you without hair, or extra eyes, or a colon? The answer is more soup. Basically, the smart people pour a special cell soup called Cas9 protein into the cell along with soup made of RNA that tells it what part of the DNA to cut. That RNA is made of like 20 base pairs of chromosomes. If it helps, you can think of somebody's genes as a music playlist, with each song representing a base pair in your DNA. Many people have different musical tastes, but some things are just plain bad. Say there's an Alanis Morissette song on your playlist, or some other country atrocity. You don't want that anywhere near your speakers, right? You know that song is going to harsh your vibes for the rest of the day, so what you do is you use a specific command to cut it from the playlist, and you're careful to not cut the good stuff next to it because nobody likes losing their earlobe. Unlike humans, though, cells and proteins don't have brains to tell them, no, keep that one, I'm going to use that later. So they need a guide RNA to tell them to only remove the sucky DNA, not the rest of it. Of course, this means that protein production gets messed up, and all the other ways that smart people decided to mess with the instructions for writhing piles of cells just made the soup go bad, because the instructions just weren't right anymore. This new one doesn't, because it knows how to tell other things how to do things right the first time. So now, your sucky DNA is gone, and you're free to live your life without anything hindering your, life. Now, I know you're saying, this is all nifty, but why do I care? Well, the thing is, CRISPR is actually pretty common nowadays. Some smart people from America use CRISPR to completely annihilate HIV strands in some pretty rad lab rats. Okay, maybe not completely annihilate, but still, CRISPR was used for some pretty awesome stuff, medically speaking. They used CRISPR to cut away HIV strands from an infected cell, all without harming any other DNA. The actual good, harmless, tasty soup stuff. But how can this possibly happen? Well, in order to deliver CRISPR and the wonderful Cas9 soup to a massive infected cells, you've got to get it past the wonderful immune system, since, it's kinda twitchy and likes to attack anything that isn't itself. In order to do this, you've got to pair it up with an AAV, or an adeno-associated virus, which is basically another pacifist that doesn't do anything harmful or replicate unless another virus helps it. So, you've got your load of soup, CRISPR, and an AAV. The happy trio make their way into the cell, replicate, and then start beating the living daylights out of HIV strands. Oh right, there's also the other kind of CRISPR, the boring one that doesn't have any sciencey applications for evil scientists. Basically, as we said, cells don't like visitors around them, so in their instructions, and in their walls, they stick parts of their visitors' corpses so they'll see them coming next time. I guess that works. They put the corpses in their instructions really neatly, though, after palindromes that aren't words, but cell text. It's weird. Cells are weird. You're weird. And how do we know all this stuff? Science. We could make a religion out of this.